Hi everyone. In this lecture we're going to go through chapter 10, which there's a lot of information in chapter 10, so make sure that you cover this chapter thoroughly in your textbook. What we're going to now talk about is, now that we understand the structure of DNA and what DNA is and the letters, the A's, T's, C's, and G's and what they mean, now we're going to talk about how we turn those letters into an actual product. So this particular lecture is going from our sequence of DNA into a physical thing, which happens to be proteins. So we're going to talk about how we get from a gene to a protein. It involves two major steps, which we'll cover in this lecture: transcription and translation. We're also going to talk about the universality of the what we call the genetic code, and how every organism essentially is using the same system. And then finally, we'll finish off by what we do with a protein once we're finished assembling it. All right, so the part of the human genome which is encodes for proteins has a special name, and that name is called the exome. So remember, your entire genome would be all of your DNA base pairs on all 46 chromosomes. But the part that we're going to be looking at today is just the exome. That's only the part that codes for proteins. And remember, in our very first lecture, how we talked about how there aren't a whole lot of genes. We have about 23,000 genes as humans, but that was small in comparison to other living things. If you remember, rice had over 50,000 genes just for a rice plant. So we have a sort of a compact genome. A lot of genes fit into a very small area. So most of the rest of the genome actually is controlling protein synthesis. So we're talking about when to do it, how fast to do it, which cells to do it in. So the rest of our DNA is involved in our overall instructions on how to build and maintain a living thing. It's just that those other pieces outside of the exome are more for regulation and control. All right, so how do we make a protein? So we've got a set of instructions that is in our DNA, and it's coded for in those letters that we've learned about: the adenines, guanines, thymines, and cytosines. It's a process we refer to as protein synthesis, and it involves two types of nucleic acids. The first type we've already talked about, which is DNA. We're going to introduce a new type of nucleic acid today, and it's called RNA, which stands for ribonucleic acid. So remember, when our discussion of DNA in the previous lecture, we talked about how DNA stands for deoxyribonucleic acid. Well, here's the other nucleic acid, RNA. Proteins are the jack of all trades when it comes to what they do for us. They do just about everything you can possibly think of. So we have proteins that are responsible for structure and connective tissue. We have proteins that are responsible for things like transporting oxygen or iron, our immunity proteins, again, moving our muscles. You name it, there's a protein that's responsible or that's a part of the function for that particular cellular, again, function. That's the idea. All right, so what we do then is, as we've talked about, cells are differentiated inside of our bodies and inside of our tissues. So certain cells in certain tissues will only produce what we call a subset of genes, right? So the genes that your skin, the proteins that your skin are producing are not the same proteins that your heart is producing, or your liver, or your muscles, right? So there are different proteins that we need in different tissues in our bodies. All right, so what's interesting in our discussion today of protein synthesis is, while the set of instructions on how to make these proteins is in the DNA, it's not actually DNA that we use to make proteins themselves. We have to make a copy of that information, because if you remember something very important about DNA, it's housed inside the nucleus, and the only time it ever leaves is when the cell is going through cell division. But when we actually need to do the rest of the regular functions that a cell does, we need to have access to that information. But the DNA can't come out of the nucleus. So what we have to do instead is we have to make a copy of that information. And that's where RNA comes in. RNA is the copy of the information we need to actually physically make those proteins. So there's some important differences between RNA and DNA. And let's talk about them for a few minutes. DNA is double-stranded. Okay, we learned that. Deoxyribose is the sugar that we use, and it has the bases A's, T's, C's, and G's. RNA is different. It's only a single-stranded molecule. We only need one copy of that information. We don't need a double-sided copy. We just need a single-sided copy, so we only have one strand. It uses a different sugar, which is ribose. So if you're looking at the two sugars, the difference is right here. Ribose has an oxygen here, and deoxyribose does not. That's the difference. The other big difference, and the one that's more that you'll remember probably a little easier, is that there's no thymine in RNA. So T doesn't exist. Instead, we have a new base called uracil. So there's uracil, cytosine, adenine, and guanine. But uracil behaves the same way that thymine did. It still pairs with A's. So in RNA, rather than A's pairing with T's, A's pair with U's. So it's kind of a new rule to remember. There are three major types of RNA, and each one of them is involved in the process of protein synthesis. We're going to talk about messenger RNA, ribosomal RNA, and transfer RNA. So let's talk about messenger RNA first, or mRNA as it's abbreviated. And this is the type of RNA that actually contains the information on how to construct those proteins. So we make RNA, messenger RNA inside of the nucleus, and then it leaves the nucleus and it goes out to the ribosomes. I don't know if you remember, but in our cell discussion, we talked about these little tiny organelles called ribosomes, and they were workbenches. They assembled proteins. That's where the message needs to go. So what we use actually is we look at the genetic code and we break it down into triplets. Okay, so 3 letters at a time. And each of those 3 letters is kind of like a word that we know how to read. So our bodies know how to read those words. And each of those words is referred to as a codon. And those codons tell us exactly which amino acids to assemble together. So again, kind of going back a little bit, if you remember, proteins are molecules that are made up of different types of amino acids. And the order that we put the amino acids in makes up different proteins. So most of our messenger RNAs are anywhere from 500 to 4,500 bases long. It just depends on how big our protein is. The next type of RNA we're going to talk about is ribosomal RNA. This is rRNA. This is actually what we're using to make up, along with proteins, we put all of these together and make our little workbenches where we're going to actually assemble our proteins. So this is kind of what it looks like. So you've got a large RNA base and a smaller RNA base, and it makes, again, anywhere from 50 to 30 proteins and are involved here, and it makes this little workbench that we're going to use to assemble our proteins at. Last but not least are transfer RNAs, and these are small. They're only about 75 to 80 bases long. But transfer RNAs have a really important function. They are like taxicabs. Transfer RNAs have what's called an anticodon, and the anticodon matches perfectly with the codons from the messenger RNA. So what the transfer RNAs do is they are taxicabs that pick up amino acid passengers, and they bring them to the site of assembly, protein assembly, and they drop them off. So again, it's kind of like just dropping off a passenger. So here's our transfer RNA. So here's where it'll pick up its amino acid passenger, and here's where it will bind with our codon, so it knows that we're dropping off the right passenger, right? We don't want to make a mistake and drop off the wrong amino acid passenger here. So we're going to make sure we drop off the right one. So again, in the process, which we're going to see in just a minute, so I'm kind of getting ahead of myself, we've got our messenger RNA here and our transfer RNAs here, and here's where the amino acids are attached. But we'll come back to this figure in a minute. Okay, so again, important rule to note when we're making RNA, we've got slightly different base pairing rules. A DNA T still pairs with an RNA A. That still works. C's and G's still pair the same way together. All of that still works. We've got a new rule, though. Whenever we see a DNA A, there are no T's in RNA, so we have to put a U instead. That's another one of the ways that our cells can tell the difference between RNA and DNA, is because RNA has uracil and DNA does not. Okay, so the flow of genetic information then goes from DNA to RNA to protein. So that's kind of, it's called the central dogma. One of the central dogmas of biology here is this idea that we have these processes where we're looking at our DNA inside of our nucleus. A process called transcription occurs in the nucleus where we make a new RNA molecule, okay, using DNA as a template. The RNA leaves the nucleus and goes out into the cytoplasm, where we go to ribosomes, and in the process of translation, we actually turn those instructions into a physical product, a protein. Okay, so transcription, translation. Transcription happens in the nucleus. We make a copy of a gene using RNA. Translation occurs in the cytoplasm. We take the message, we take it to a ribosome, we read the message, and we actually make a protein. All right, so transcription, again, we're inside of our nucleus because that's where our DNA is housed. We have two strands, what's called the template strand and the coding strand. Doesn't really matter. All you need is one side of the DNA in order to know what the message is, right? That's the beauty of complementary base pair rules, is you just need one half of it. And we use an enzyme called RNA polymerase. But here's what it looks like. Okay, so here is our gene. Okay, we're going to unwind and pull apart the DNA exactly at this location that we need. And then we're going to use this template strand, and we're going to begin to bind free mRNA nucleotides, and we're going to put them in the appropriate order based on, again, the message that we're reading here. And again, every time you see an A, you have a U. So all of the other base pair rules still hold, except that whenever we have adenine, we use uracil in our messenger RNA instead. Next comes translation. So we're going to take our message, and we're going to take it out to the ribosomes. And in the ribosomes, we're going to read those letters. Remember, the words are 3 letters at a time. Those are called codons. We're going to read the codons. We're going to call in a matching transfer RNA that has the matching anticodon, and the transfer RNA is going to drop off those amino acid passengers. All right, so let's take a look at it here. So here is our sequence of messenger RNA, and there's something called a leader sequence here. The leader sequence is a non-coding region of messenger RNA, and it basically just tells the ribosomes to become assembled. Because remember, the ribosome is a small subunit and a large subunit, and they actually have to assemble. So what happens is this leader sequence here tells the ribosomes, hey, we're going to start making a protein, get ready. So the small ribosomal unit subunit is brought in, and then we always have a universal start codon. So every single protein that we make, all 23,000 of them or so, are always start with AUG. It's the universal start codon, and it basically tells our cells it's go time. Let's start making this protein. So AUG happens to stand for the amino acid methionine, which you don't have to know necessarily, but you should know that there's a universal start codon. And once we see that, the large subunit is going to come in, and we're going to continue moving along this line, and we're going to continue to read these codons. Again, they're triplets, right? 3 letters at a time. And then we bring in the next amino acid. And then these transfer RNAs will drop off, and we'll begin to build this chain of amino acids. Okay, let's do a quick comprehension check. Blank produces RNA from a DNA template, and blank produces protein from an RNA template. Okay, so think about that. What produces an RNA? Which process produces RNA from a DNA template? And which process produces protein from an RNA template? Is it replication transcription, transcription translation, translation transcription, or transcription and replication? It's number 2. It's transcription and translation. So transcription, if you think about the word transcribe, when you transcribe something, you're writing something down. You're sort of making a copy of what you're hearing. We're making a copy of DNA using RNA, right? And in translation, when we're translating, say, from one language to the next, right? We're reading words in one language and then translating them into something that we can understand. Kind of same thing in our cells. We're translating letters that we're reading and turning them into actually products. All right, let's talk about the genetic code real quick. So the genetic code is great because we figured out what all of these words are. So these triplet words, these codons, that's what's in our DNA. We know what they mean, and we know exactly which amino acid is called for by which codon. Okay, so those 4 letters, A, U, C, and G, we can combine them in 64 different ways. There are 3 stop codons, so just like we had a universal start codon, we all start with AUG, there's 3 different ways to call for stop, to say don't make any more of these amino acids. And the table, which I'm going to show you in just a minute, actually let me show it to you right now, and then I'll talk about those other 3 points in just a minute. So here's our amino acid table. And the way that you read this, which is very important because you're going to need to know how to do this, you find the first letter of the codon on the left, you move to the second letter, which is here on the top, and then the third letter is going to be here on the right. So let's do an example here. Let's take CCG. All right, so let's find the amino acid for the codon CCG. So starting on the left, I'm going to find C. Here it is. Then I'm going to move over to the top here. Here's my next C. All of these are C's. And then on the far right-hand side, I'm going to find G, CCG. Here we go. CCG happens to code for proline. Okay. 3 important things to know about the genetic code. Number 1, this code is universal. Every living thing is using the exact same code. So there's not a different code for plants or animals or bacteria or fungi. We're all using the exact same code. We all use this. Number 2, there is redundancy in the code. Okay? Redundancy means that there are multiple codons that will code for the exact same amino acid. So there are 4 different ways that we can ask for proline, right? And there are 4 different ways we can ask for glycine. So there's redundancy here. The next important point is even though the code is redundant, it is not ambiguous. A codon can only code for a single amino acid. It will not code for more than one. So if I say CUAU, I always know it's tyrosine. It will only be tyrosine, and it will never, ever, ever be anything else. So we never have to try to figure out, I wonder if they're asking for serine or tyrosine. Nope, we'll always know exactly which codon it is. So going back to the previous slide real quick, it's redundant, it's not ambiguous, and it's universal. Okay, last thing I want to mention here as we wrap things up is a string of amino acids is not necessarily a protein. It's just a string of amino acids. We actually need to fold proteins to give them their appropriate function. So we actually have, technically not all proteins will go up to quaternary structure, but there's technically 4 different levels of structures we can have for a protein. So your primary structure, again, is just a chain of amino acids, which is great, but it doesn't do anything. Next, we move into secondary structure. We have 2 different types of secondary structure. We actually have something called an alpha helix, which is like a coil. And then we have what are referred to as these beta-pleated sheets. Okay, so all proteins will go from a primary structure to a secondary structure to what's called a tertiary structure. In the tertiary structure, we begin to take the alpha helices and the beta-pleated sheets, and we begin to fold them up in on each other to make a 3-dimensional shape. Some proteins will move on to have a quaternary structure where you have multiple proteins here. So this is 4 different proteins that have come together to create one structure. Not all proteins do that. What's interesting is that, just like anything, mistakes can happen. And mistakes in protein folding can and do occur. And usually it's because there was a problem with protein synthesis, or maybe it was a problem with the protein itself as it was folded. This can actually lead to disorders. And there are actually several very well-known disorders that are caused by these misfolded proteins basically accumulating in our brains. So they sort of stop functioning, and they're just sitting there clogging things up in your brain. And so if you're familiar with some of these disorders like Alzheimer's, Huntington, Parkinson disease, these are all responsible. What's causing them is actually these misfolded proteins. And these misfolded proteins are just sitting there accumulating in the brain. And as they can accumulate in the brain, they begin to interrupt brain function. And that brain function then is responsible for so many other parts and things that we do in the body, of course, until you see consequences related to those diseases. All right, last quick comprehension check. A codon in messenger RNA consists of how many nucleotides and encodes for how many amino acids? 3 and 1, 3 and 3, 1 and 3, 1 and 1. It's 3 and 1, right? It's a triplet code, and it codes for 1 amino acid. Okay, important terms and concepts to know.